studying Prescott's microbiology after a long time. So, not a long time, I mean, it's been a month, right? So, I'm not sure if my streams will be regular from now on, but then, I think I will update the next time I disappear like that. So, for today's topic is the continuation of the chapter number 3 where we left off, and that is the protein secretion pathways in prokaryotes. Guys, so today's topic is a little bit complex, and I think it's just a little bit straight from the from the normal microbiology stuff, right? So before me going to that, let me just check if my stream is working, because it's been a long time since I streamed, and I don't know if the settings I put is working fine or not. So just let me check it. Check. Oh, okay, so it is working fine. Okay. So now we turn off this, and just let me check the OBS first one more time. Okay, so nothing is wrong. So without further ado, let's start our work today. So my pattern is first telling me what is protein secretion and what are the major things that we are going to study, and for what kind of creatures are we going to study these secretion pathways, etc. Look, this is what we are going to study today, and why do we study it. And so yeah, I will just explain it in simple terms, just the way it is written in the book, and maybe more simplified. So my pattern is just first explaining the diagrams and the basic stuff, right? The bold stuff, the apparent stuff, and not going into the detail like the examples and stuff like that. We will do that after we do a read through. And by the way, I do this kind of things when I want to make you understand, okay? So don't get bothered with it, okay? Exactly. So first of all, we are studying protein secretion pathway. Protein secretion pathway in prokaryotes. First, prokaryotes, we all know that these are the single microorganisms, single-celled microorganisms. They don't have true nucleus. Well, I guess prokaryotes can be multiple, but they don't have true nucleus. Now, their major functions, revolves around producing the proteins. So they need a mechanism first to produce those proteins, and for that they have ribosomes, okay? But then after they produce protein, I mean, some remains inside the cells, right? Some remains inside the cell. They don't need to be transported outside. But then there are enzymes, like extracellular enzymes. There are toxins, and there are chemicals, and there are minerals or materials they want to secrete out of the cell, right? So for some of them there are channels, right? But for protein this is different, because protein is very sensitive to pH and stuff like that, and there are so many enzymes in the cell that they are prone to degradation, so they need special treatment for their transportation, special mechanism. So protein secretion means we are secreting the protein outside of the plasma membrane, or we are letting it reach to the plasma membrane. Oh my god, what was this again? Man, I don't like to get disturbed when I study, what page were we on? Like, do I know what page we were on? Was it 81? Not far away. Yeah. So we secrete it out. So by the way, remember, in prokaryotes we have gram-positive bacteria, gram-negative bacteria, and the archaea. So for our archaea, we don't know the pathways that much, clearly, because archaeas are just recently, by the way, this book is old. I don't know how many progression has been done in the recent years. But archaeas, they are saying that we don't know much about the secretion pathways. While the gram-positive and gram-negative bacteria, we do know. The gram-positive bacteria don't have much complex pathways, in comparison to the gram-negative ones. And why is that? Because the gram-negative cell wall is pretty complex. To make it to secrete it outside of the gram-negative cell walls, it is quite a work, because gram-positive have only peptidoglycan, while the gram-negative actually has, the peptidoglycan, the lipopolysaccharide layer, and again the oh my god, lipopolysaccharide layer. Like it is sandwiched between these, and so it is difficult. And by the way, gram-positive has a lot more bigger pores and more in quantity pores than the gram-negative. It is way more compact, so it doesn't allow materials to go outside. So this is the general idea. We want the proteins to go outside of the cell. We want to secrete it out to the cell, and for that we need other materials, and so it sets up a pathway for us, so we can secrete that protein out. Now for general, we have sex dependent pathway, right? Sex dependent pathway. By the way, so today we are going to study the bacteria one, okay? Because archaea one is, not that much being discovered, and it is there is a one pathway, that is ABC pathway, and yeah, it is present in all of these gram positive, gram negative, and the archaea ones. So the important one, the most important one that you need to know first is the sex dependent pathway. Sex dependent pathway is for both the gram positive bacteria and for the gram negative bacteria. The name shows that there is a material, a thing, a protein or anything that is known as sec, right? And that pathway is dependent on it. So we have kind of proteins that are in short sec, and they are A, B, C, D, a lot of different types. And the pathway is dependent on that for us to transport the proteins. Now sex dependent pathway transports only the proteins that are not folded, that are not matured completely, okay? Like the structures of the proteins, the primary structure, the secondary, tertiary, quaternary structure. So tertiary and quaternary are quite mature structures. So before going into those structures, there are chaperones, the proteins that keep the protein as it is. It is a protein that keeps other immature protein into themselves, either to mature them or either to retain their state as it is. So in this case, the chaperone that we are going to use are the only ones that retain their original state. Like they are in immature state, and so they let it keep it that way until they are left out of the plasma membrane. So anyway, so this is the sex dependent pathway. It is present in both gram positive, gram negative for the secretion of protein. It is the general pathway, okay? So the mechanism we will study after I tell you the other names. Now in gram negative bacteria, we do have sex dependent pathway, right? But we also have 5 other mechanisms for them, and that is the type 1, type 2, type 3, type 4, type 5. Now there are 2 pathways, type 2 and type 5, that is dependent on the sec pathway, right? The sec dependent pathway that we studied, they are dependent on this one, the sec one, while there is one, the type 1 and the type 3 one, right? They are independent. They can do their own work on their own. They don't need the sec one, right? Like these. Now, and there is another one, the type 4 one. By the way, these are all occurring in gram negative bacteria. Now there is a type 4, and it is quite a switch. It usually is sex dependent, but it can be independent, like it's upon their mood, whatever it wants. So yeah, the other ones directly transport the protein outside to the plasma membrane, these connected structures, while in the case of the sex dependent, by the way, chat dependent is coming, but we don't want to discuss it right now. It is not explained in this book, so we will just Google it at the end of the stream. What was I saying? You see, these two are not connected, the type 2 and type 4, because it has a space in it. Means they first have to go into this periplasmic space and then to another channel. While in their case, they just go directly to the another channel. They don't have to be there in the periplasmic space. So yeah, this whole thing, they just explained everything. So we will just read it at the end. So first, now let's go back to the sex dependent pathway, right? We talked about the sex dependent pathway, so let me explain it with the help of diagram. First, remember, we said this is a protein secretion pathway. So first we need a protein. Protein is coming from where? It is coming from ribosome. but we take the protein from the ribosome, it's in immature form. We call it the pre-protein. Here you see, wait, let me push the diagram to the side so you can see. My avatar, I think, is blocking it some way. So this is the pre-protein. See, this is in a linear form, this is not folded, and we know that the amino acid, it is made from the amino acids. The amino acids have COOH at one side and NH at one side. Now remember, we have a signal, a signal that this protein needs to be secreted out of the plasma membrane, and that signal is usually attached to this area. There is a sequence of amino acids, I think it's 20. It's 20 or something like this, 20 amino acids that is arranged in a coded form that acts as a signal to attract what? How will it go to plasma membrane? Of course, it doesn't have feet. It doesn't have locatory, motory, anything that it can go outside. So it needs a helper for it to carry them. So first, the helper before a helper comes. So in this case, the helper is Sec-A. Remember, this is a motor protein that helps carry stuff. But this Sec-A has a requirement, and the requirement is that it only recognizes Sec-B. So how will the Sec-A attach to this protein and carry it to the plasma membrane? So, and that because it just only recognizes the Sec-B. So that means we first need to bind this Sec-B to the pre-protein, and we bind the Sec-B to pre-protein. And how does Sec-B binds to the pre-protein? It is because of the signal, okay? So because it recognizes the signal, it attaches to the pre-protein. And because Sec-B is attached, now Sec-A comes and attaches to itself. Now for now, by the way, Sec-B is a chaperone protein. It is a protein that keeps the pre-protein shape as it is. It doesn't let it fold. It doesn't let it mature. And because we don't want it to be folds to be attacked by, enzymes and etc. We want to preserve it so it can go and change into its environment, outside, wherever it wants to go. So till now we understand that, right? By the way, it isn't attached to this. This is happening in the cells, like here. This is our protein, this in this shape, and there is Sec-B, and it attracts the signal and it goes there. So then we have this protein of ours, and then we have Sec-B. Then Sec-A here, it says, oh, I see Sec-B attaches to something. So it goes towards here, and then we have this complex, Sec-B, Sec-A, and this. Now this is plasma membrane, right? Now plasma membrane has other Sec proteins that act as a channels, you do get it, channels, right? That allows you to transport stuff with a semi-permeability. So we have a Y-Sec, a E-Sec, and a G-Sec. Collectively we call it YEG-Sec, right? See here, it is written here. Now we have these three channels. Now that Sec-A binds to this B and our pre-protein. Now it will want to transport it to the channels, because what's the point in lying in the cell when they have already the machinery ready, and because Sec-A has the machinery to produce energy and let it carry, because it is a transporter protein. So this Sec-A, wherever it is in the cell, it goes straight direct towards the Sec-YEG complex proteins, okay? So it goes there, and after it goes there, as you can see, this amino acid was first at this side, and after binding with this YEG, it is moving outside of the plasma membrane. I hope you guys get what I was saying, right? So this is generally the Sec-YEG pathway. What happens? We have our pre-protein from the ribosome. Sec-B, a chaperone protein, binds to it and let it rest in its pre-protein form. And then Sec-A recognizes the Sec-B complex, and it says, let me bind with it. So it comes and binds with it, and then it transports towards the Sec-YEG, and so our protein is secreted outside the plasma membrane. This happens both in gram-positive and gram-negative. So I hope till now we understand what we were saying. Now the next thing is we have type 1, type 2, type 3, type 4, and type 5. It looks scary, right? It isn't. So don't worry, and let me say what is written here. First of all, don't be scared of these things, right? Like, this looks a lot of work. This is just ATP converting into ADP. Why? Because it is working. It needs an energy source. So when these channels work and when this transportation works, it it needs ATP. It usually needs ATP, but sometimes it uses the proton motive force. Now, what is proton motive force? It is the cell gradient. Remember that we had the, just think about the H positive ions, here, like this. And we have, for example, let's make it plus, okay? For example, these are hydrogen proteins. Now at this side, the positive charge is a lot, quite a lot. This side protein is a lot. And this side, after outside the plasma membrane, there is not much protein. So these proteins, not proteins, these protons will want to go to the other side. And so here there are so many positive charges, and it is in stored form. So this is a kind of potential energy. Remember potential energy, the one that is in stored form. So when it moves and it turns into kinetic energy, so we can use that to help transport our protein. So we do use sometimes the what do you call it, the proton motive force as well, which is the use of the potential energy of the different gradient across the plasma membrane. Now remember what I said, we had 5 and we divide it into three parts: type 1 and type 3, type 2 and type 5. These two are dependent on sec pathway. See, the arrows from sec goes here. This one we will talk about later, because this is semi-dependent. It can be dependent and it cannot be dependent as well. So that's why the arrow goes there as well. Well, if looking at this side, they both don't need any sec pathway. The protein directly go towards the type 1 and the type 3. Now, by the way, the scary one is the type 3 one, because this one causes so much damage. The protein that this type transport are much scary. They produce toxins, they produce chemicals, they secrete chemicals, protein chemicals, that damages our host cells and our nervous systems, and stuff like that. It is pretty damaging, but it has a lot of important stuff other to do as well. It doesn't only transport protein, it can transport glucose, etc., right? So I think this one's pretty complex one. This one's the complex one among all. It's said below, right? So how does this mechanism work? So I haven't studied what is this tool C, what is YOP, and what are these things, the pol S and stuff like that, YSCG. It's just a representation, it's the protein names, right? Just as we studied the sec-YEG here, it's the protein name. So yeah, just don't go, I think, yeah, these are protein names. So anyway, these two forms channels, while these two don't form channels, and they are sec dependent pathways. Now what? Protein goes into chaperones, either go it into or either don't go into it. It depends if you want your protein to be folded or if you don't want your protein to be folded. Like here, in this case, this is still a pre-protein. In this case, it is still a pre-protein, meaning it is not mature and it is in its linear form, where it is not still folded and have bonded. Now there are two types of chaperone proteins. One chaperone protein let it in the state of being infolded, immature form, while there is another type of chaperone protein that let it mature in the cytoplasm, so in a protective way to mature itself. So I think type 1 and type 3 secrete directly matured proteins, while sec type 2 and type 5, not sec 2, type 2 and type 5 only transports the immature one. So there's another one. No, I think they don't. Wait. I think only the type 3 one transports the folded one. Well, I guess I'll have to search it later. We will search it in the end. So this is important, so before reading anything else, let me read this. this paragraph. Gram-negative bacteria use type 2 and type 5 pathway to transport proteins across, why is it lagging? Across the outer membrane after the protein has first been translocated across the plasma membrane by sex-dependent pathway. Okay, so they are just saying that the type 2 and type 5 are used when already the protein is transported by the sex-dependent pathway. By the way, remember that the sex-dependent pathway only goes towards the protoplasm, okay? Not the protoplasm, the plasma membrane. The sex-dependent pathway, for example, this is plasma membrane and this is our cell wall. Always remember that the sex-dependent pathway only goes on to the plasma membrane, right? While this one, the cell wall, for us to go here or outside of this, we need other type of pathways, and they are type 1, type 2, type 3, type 4, and type 5. By the way, type 1 is also known as ABC pathway. Type 1 protein pathway here. Which one was it? Yeah, this one. This one is present in every prokaryote, like in archaea, in gram-positive bacteria, and in gram-negative bacteria. They are all present. ABC pathway, okay? Why? Because it uses a lot of ATP. And it transports a variety of material, like it is the basic transportation system of all of them. So I think we should just start reading now. It will be done in a minute. So how about we relax for a second first with how do you like it? Yeah. Let's relax. Is this much relaxation fine for us? Yeah. Yeah, I guess this much is fine. Now let's just read. The membrane of the prokaryotic cell envelope, that is the plasma membrane of the archaea and gram-positive. Man, I can't read. The membrane of the prokaryotic cell envelope, that is the plasma membrane of the archaea and gram-positive bacteria, and the plasma and outer membrane of the gram-negative bacteria. They are saying that there are membranes, the cell envelope that is present in the archaea and the gram-positive bacteria. They preferred the gram-negative bacteria as a separate because its cell wall is complex, so the transportation mechanism is complex as well. So, and they are saying that they present a considerable barrier to the movement of larger molecules into and out of the cell. So they are talking about the proteins. By the way, when we study the sex-dependent pathway or the type 1, type 2, type 3, they only talk about it going outside. We don't talk about it going inside, because I think for the inside there are different channels. How are large molecules formed, which some of these structures are made transported out of the cells for assembly? Now they are saying how, there are structures outside of the cells. For example, in the peptidoglycan layer, in the cell wall, there are a lot of structures, enzymes that works for binding to other cells. How are those structures formed? How are proteins for those structures carried out? And of course, these are by the help of this protein secretion pathway. Furthermore, exoenzymes. Of course, we talked about enzymes. Exoenzymes and other proteins are released from prokaryote into their surrounding. How do those proteins get through membranes of the cell envelope? How do they get out? Clearly, prokaryotes must be able to secrete proteins. The research on protein secretion pathway has been mushroomed in the last few decades, in a part because of the fundamental importance of the protein secretion, but also because certain protein secretion mechanisms are common. What's wrong with this? Can be exploited for vaccine development and a variety of industrial processes. Now people will be saying, why do we study how it transports, etc. I mean, first is general curiosity how it works. But by studying how it works, we can use their pathways and exploit how they secrete or do some stuff, so we can use the vaccine. Like for example, think if there is a channel that secretes a toxin, and we want to kill this bacteria or make them unworkable. So we will produce the drugs, and since we know that this channel or this pathway is the cause for problem, so these drugs should attack this area, and this is only possible because we have studied how it works, how the secretion system works. If we didn't know how they are secreted, we might not be able to form a drug that could attack it and so cure disease. Furthermore, an understanding of protein secretion can be explored. Okay, because relatively little is known about the archaeal ones. Archaeal protein system. So this section will only talk about the gram-positive and the gram-negative. Now, what is general overview of the bacterial protein secretion? Protein secretion poses different difficulties depending on whether the bacterium is gram-positive and gram-negative. There is gram-positivity and gram-negativity is you can say pretty vast and important concept in the bacteria. Just consider them as separate things at this point. You know how important they are. In order for the gram-positive bacteria to secrete proteins, the proteins must transport it across the plasma membrane. Gosh, I can't even read and speak or what. Once across the plasma membrane, the protein either passes through a relatively porous peptidoglycan into the external environment, or it becomes embedded in or attached to the peptidoglycan. After we secrete the protein, it can either embed into the plasma membrane, it can either embed into the peptidoglycan, or it can go to the external environment, or it can remain in the periplasmic space for how long it wants. Gram-negative bacteria have more hurdles to jump when they secrete proteins because it has a stupid layer of the cell wall on it, a complex one. They too must transport the proteins across the plasma membrane, but in order to complete the secretion process, the protein must be able to escape the attack from protein degrading enzyme. When we are secreting the proteins, of course there are a lot of enzymes that degrade proteins. For example, proteases, pectinases. So these kinds of enzymes are dangerous for specifically the pre-proteins, the immature protein that haven't even been folded, that are not resistant to the enzymes. So they must be transported across the outer membrane in a protective way. In gram-negative bacteria, protein can be transported across the outer membrane by several different mechanisms. Some would be bypass, the sec-dependent. Bypass means it doesn't stop on the periplasmic membrane, it goes directly outside. Or you can say some first go towards the sec-dependent pathway, and then afterwards that another pathway. All protein secretion pathway described here requires the expenditure of energy at some step in the process. We know. We take it from ATP, GTP, and protein motive force. Not protein, proton motive force. I already told you what the proton motive force is. And GTP is guanosine triphosphate. This one is adenosine triphosphate, as you guys would know. Now the sex-dependent pathway, sometimes called as general secretion pathway, it is highly conserved and has been. Wait, let me search. What is the complete form of the sex-dependent pathway? Completely forgot. It was a name of some protein. Sex-dependent pathway. Or is it just simply secretion dependent? What's its full name? Full name, man. The Google doesn't. Full form. What? What is sec in sick? Wait. They don't answer when I ask like this. You guys can see it yourself as well. Wait. You guys cannot see it. Why? Here. Now you guys can see sex-dependent pathway. What is sec in it? What is this, man? What is wrong with it? What is sec in it? refers to a set of bacterial protein and a pathway. Gosh, I just want to know what the sec represent in sec pathway. Excuse me? Excuse me? Should I just ask the ChatGPT? What is SEC in secretion pathway? Okay, forget it. I don't think we should. Red pathways. Okay. Let's forget it. I'll search it at some other point. This is not funny. Wait. Okay, let's continue. It translocates protein across the plasma membrane or integrates them into the membrane itself. Proteins to be transported across the plasma membrane by this pathway are synthesized as pre-secreted proteins, called as proteins. Mom, can you take these? Excuse me? Excuse me? Hey! Mom, can you take these? I'm disturbed by this voice. No. I have 10 minutes more, I guess. The protein has a signal peptide. Guys, we all talked about this. Why is it boring? They are saying that this signal attached to the sec pathway. Amino terminus, you know? What? C terminus. Right, C terminus, right? Amino terminus. Don't tell me they are talking that it is attached to the NH terminus. NH3 terminus. Okay, must have been wrong. Maybe in sec-dependent pathway the attachment is on the N terminus, and in the TAT pathway it is on the C terminus. By the way, C means COOH, okay, which is recognized by the sec machinery. Soon after the signal peptide is synthesized, special protein called as chaperone protein, or aka sec B, as we said, binds to it. Pre-protein is binded to sec B. This helps delay protein folding, thereby helping the pre-protein reach the sec transport machinery in the conformation needed for the transport. Means it is easy for us to mold it into the machinery. Like, you know, if you are big and there is a small car and we need to put you in it, right? But if you are water, you are easy. You know, if you don't have a definite shape, we can, you know, put you into the stuff as we want. So there is evidence that the translocation of proteins can begin before the completion of their synthesis by ribosome. Oh, okay. Certain sec proteins, sec Y, E, E, G, A, are thought to form a channel in the membrane through which the pre-protein passes. Another protein, sec A, binds to the sec Y, E, G protein and a sec B pre-protein complex. Okay. It binds B and then B with to the sec Y, E, G, and it acts as a transporter and energy provider. Motor. Yeah, to translocate the pre-protein, but not the chaperone protein. Oh, I see. Guys, they are saying that when it attaches to the sec B, it only helps this pre-protein move along the sec Y, E, G, and it lets the sec B here. You know, sec B is attached to N terminus, right? It was attached here at first, but after sec A is bind to the sec Y, E, G, it allows this NH to leave this sec B and slowly moves from sec Y, E, G outside of the, you know, plasma membrane to the periplasmic space until they reach the end point. So I see. Now after the pre-protein emerges from the plasma membrane free from chaperone, an enzyme called as signal peptidase removes the signal peptide. Because we don't need it in our protein structure, it is rubbish. The protein that folds into the proper shape and disulfide bonds are formed when necessary. Protein secretion in gram-negative bacteria. They were saying that it just folds however they want and the bondings just form however they want. I mean, the way it is designed. Protein secretion in gram-negative bacteria. Currently, 5 protein secretion pathways have been identified in gram-negative bacteria. Recall that the gram-negative bacteria have a second outer membrane that the proteins must cross. Remember? Of course, we remember it. So the gram-negative one uses type 2 and type 5 after the secretion sec-dependent pathway, and they use type 1 and type 3 directly when the protein is in the cell, okay? And type 4 it just acts however it wants. It can be, it usually is, it is linked to the sec-dependent pathway, but it works on its own, it works both ways. Now, type 2 secretion pathway is present in a number of plant and animal pathogens, including Arena, Caryophyllora, Klebsiella pneumonia, and the Pseudomonas aeruginosa, and the Vibrio cholera. So they are saying that it is present in the pathogens. I think pathogens need more secretion pathways because their proteins are, deadly. It is responsible for secretion of proteins such as degradative enzymes, cholineases. Guys, this is a special form of the glucanases, I think. Wait, I did search it. Do I have to search it again? What was it again? I forgot. Oh yeah, that breaks down a pool and a polysaccharide. A simple single chain. Oh hey, Rampukar Singh, you are quite. So let's continue. Cellulase, pectinase, protease, and lipases. It is responsible for secretion. Means that they are secreting the protein that are dangerous, for the host cells. Of course, not for themselves because they are already secreting them. Now, type 2 systems are quite complex and contain as many as 12 to 14 proteins. I could repeat what? Which one are you talking about? Just the type 2 protein secretion pathway or what? Which one? I don't know. So we are just talking about the secretion pathways. Oh, okay. I'll just tell you from the type 2 protein secretion pathway. This is the second type that we are studying. No, by the way, this is the first one that we are studying in the order. I already explained how they work, so I'm just repeating what these lines are saying. So for type 2 secretion pathways, they are sec dependent. Oh my god. They are sec dependent pathway. No, I don't feel demotivated. It's been after a month I am streaming. So yeah, I only stream when I'm in motivated mode, so I'm feeling fine. So type 2 systems are quite complex and can contain as many as 12 to 14 proteins, most of which appears to be integral membrane proteins. Guys, integral membrane proteins are those that are embedded in the membranes, okay? So they are saying that the sec protein pathway had only, I think, 5 to 6 proteins, while the type 2 system has more, 12 to 14 proteins. Even though some components of the type 2 system span the plasma membrane, they are saying most of the proteins are on the plasma membrane. They apparently translocate proteins only across the outer membrane. Even though they have proteins in the plasma membrane, like this is plasma membrane, even though they have proteins in the plasma membrane, right? But they only work to transport, outside the outer membrane. So this doesn't count as the plasma membrane. This one counts as. I think we should finish this Pukur and then we should talk about the live ones. I think it's disturbing my pace for studying. We can talk about the motivation and this kind of stuff at the end. So yeah, in gram-negative and gram-positive bacteria, any other plasma membrane translocation system called as TED pathway can move protein across the plasma membrane. In gram-negatives, these proteins are then delivered to the type 2 system. The TED pathway is distinct from the sec system in that it translocates already folded proteins. Guys, the TED pathway is like, the sec-dependent pathway, okay? But the only difference is that the sec-dependent pathway transports the not folded proteins, and while the TED one translocates the folded proteins. Now the type 5 protein secretion pathway, our most recently discovered protein secretion system. They too rely on the sex-dependent pathway to move protein across the plasma membrane. However, once in the periplasma space, many of these proteins are able to form channels in the outer membrane through which they transport themselves. These proteins are referred to as the autotransporters. Autotransporter, guys, they form the channels in the cell membrane itself, and then they transport themselves as well, and they help the other ones as well after they make the channels, and then they just disappear after transporting. Other proteins are secreted by type 5 pathway with the aid of separate helper proteins. Yeah, they do need helper, but they can form channels as well. What is this? Where did it go? Wait, I told you we can talk after this ends. There is only few topics left. Where did it go? The chapter name, it is the prokaryotic cell structure and function. Gosh, I don't know where did it go. Prokaryotic cell structures and function. Wait, isn't it here written here? Yeah, it's just about prokaryotes and their whole components and how they work a little bit. What is this? Yeah, here, bacteria cell structure and function. Gosh, where did it, what's this doing? Now, there is a type 1. You jinxed it, I think. It doesn't want to study anymore. The ABC protein secretion pathway, or you can say the type, oh my god, what is this? What is this behavior? Oh my god. Hey, can you talk a little bit slow? What is, okay, let's refresh it. What is this doing? Do I have to read it? I mean, it just, what is this? It won't take time. We only have type 4 and the type 3. Like, I'm just reading. I already done the explanation stuff, but why is it moving like it doesn't want to study? Anyway, I will just wrap up with the main points. I don't want to read it and it just disappear anyway. The ABC protein secretion pathway, aka type 1 protein secretion pathway, and it is helping in the secretion of the toxins and a lot of other lipases, peptidases, and the proteases. Peptidases. Now, secreted proteins usually contain C-terminal secretion signals. Like I said, in the SEC pathway it was N-terminus, but doesn't necessarily have to be N-terminus. And in the type 1 it has C-terminal secretion signals. Whether signals helps the protein to know which way it will go, type 1, type 2, type 3, type 4, or type 5, or the sex-dependent pathway or the TET-dependent pathway. So it helps in what pathway will it take. And they are saying that they have seen the type 1 system that translocates protein. They have 77 ABC transporter. Guys, 77. Here we were talking about 12 to 14 in the type 2 system, and this one's talking about the 77 ABC transporters. And that only shows that how many channels do we have for this pathway, and that only shows that how much work does the cell take from the ABC system pathway, secretion pathway. Now, other gram-negative pathogens have type 3 protein pathway. Like I told you before, that the type 3 one is extremely dangerous. Not dangerous in a way because it works important stuff as well, but it produces the viral inspectors and secretors, like for example cytosis inhibitors, simulators of the cytoskeleton, reorganization in the host cell. Guys, if our cytoskeleton, our cell cytoskeleton is stimulated and is disturbed by the attacker, by the pathogen, just what do you think with our structures? Will our cell structure remain integrated as it is? It will just not stop. It will stop functioning because the I have no thoughts about the war. It's just ridiculous. They are attacking one another with no, with no purpose. I think I don't know what the higher ups doing. They're just killing people and themselves and their resources. They are wasting everything. Type 3 system also transported other proteins, including some of the proteins from which the system is built. Protein that regulates the secretion processes. Proteins that aids in the insertion of the secreted protein into the target cells. Now, some of the proteins is structural proteins, some of the proteins are functional proteins, while some of the proteins are helping in other proteins. So these are the three types of protein that the type 3 system helps transport. The structure of type 3 channels are slender, needle-like structure that extends from the cell surface. Cylindrical base is connected to the outer membrane. Here it is shown, this is a base. This is a base. This is the base. It is a syringe-like structure. It is connected, and this area as well is a needle-like projection outside, and it is directly connected, connecting plasma membrane and the outer membrane. This is specifically talking about the gram-negative bacterial. Now the last one. Just this paragraph, the type 4 protein secretion pathway are unique in the way that they are used to secrete protein as well as to transfer DNA from a donor bacterium to a recipient during bacterial conjugation. So this one transfers the DNA as well. By the way, this is also the sex-dependent pathway. The type 4 one is just as the type 2 one was. And like the type 3 system, the protein form, but its structure is similar to type 3. Its function is similar to type 2, other than that it transfers the DNA. Type 4 system and conjugation are, they are saying that the type 4 system will be explained more in chapter 13. So we are done with today's topic, and okay, let me talk with you for 5 minutes, I guess. So wait, if we are done with the topic, so I think we should, what diagram should I make it come out? What is this? Okay, whatever page we on. I think we jinxed something. This is 73. Okay. Yeah, I think so. Wait. Okay, let me answer your questions first. So you are talking about the war. No, I don't live in USA. And wait. Insta, I don't have. I have Insta, but it's not for studying, so I prefer not to give it to people. My only account for public is this YouTube. So are you coming here to study, or just want to discuss war with people? Alright, you have just 5 minutes, so I can end the stream afterwards, so be quick. Okay. Are you still there? This is mine, right? You remember? Yeah. So I can get rid of this like the way I teach you, I don't think you heard a single thing I was speaking to. Care to tell me what I taught today? By the way, you asked me I don't have viewers, so how do I feel motivated to do live? So I think I don't do it the way, I don't do it for viewers much, not for the monetization. I just want people to study, if they want to study microbiology, someone to study with. By the way, are you from microbiology field? And besides, the YouTube is a fun activity. It helps upgrade your presentation skills, maybe teaching skills. I don't know. Maybe someone can learn a thing or two, or maybe someone can read a book or two, since this era has, AI, etc., etc. People don't usually study books, etc. No. Okay. It's 28, so yeah. By the way, if excuse me, this is personal information. By the way, I do play chess on the Twitch. So if you want to watch the game and talk, just for fun, you can go there. But this is study channel, so I would prefer if you just listen to studying instead. I think I have a link in my YouTube page of my Twitch, I think. So yeah, I don't think you play chess or anything, but I like to play chess. So of course I want to get a job. It is in bad mood.
Oh, this one is good. The previous one, not this one. Yeah, not this one.
You must be a student for asking, do you want to get job and life seriously? I guess life that hard for you. Just study and go and come home and go and yeah. You play chess? Oh, that's great. Oh yeah, of course that's a joke. I don't have a specific routine for playing chess on Twitch or anything, but I do play. I do play it on Twitch. So sometimes if you are free and want to see if I'm alive, just go there. And if I'm alive, we can play together. What's your elo? I'm not a jobless. I am. My job is teaching you. Hey. Okay. Jobless. God, what a way to say. I'm not jobless. I have a lot of jobs. Anyway, so I think the time is up. No, sir, we don't have system of part-time jobs in my house, okay? Yeah. Anyway, this stream is going to end, and as I said, if you wanna just talk, fun or anything, just go to the Twitch when I am playing. This is only if you wanna ask questions. My elo is 800 in 10 minutes. What do you call it, Blitz? And then 1455. Oh my god, it's high. Mine is low. I think we can still play. So anyway, I'm ending the stream, and yeah. Goodbye. Where is it? Yeah.